Greetings to everyone. My name is Binon Teji, and here I am presenting my paper entitled Predicting Missing Links in Gene Regulatory Networks using Network Embeddings, a qualitative assessment of selective embedding techniques, which has been presented in Second International Conference on Machine Learning, IoT and Big Data 2021 with the paper ID number 44. And these are the agendas that I shall be proceeding with my presentation. Coming to the introduction, we have biological systems and which can be modeled as graphs or networks. And it is very important to comprehend the various interactions that take place in these biological networks. The gene regulatory networks, they're actually made up of a set of genes that interact with each other. But because of the lack of network, these links are often guessed to recreate these biological networks. So here we present the network embedding or representation learning, which is a recent development in graph or network data science that can be used to predict missing links in various networks and biological networks as well. So the graph representation learning, it actually involves the transformation of a whole original network into lower dimensional space. So that the representation in the lower dimensional space, which we also call latent space, correspond in notion of similarity that actually resides in the original network. Suppose we have an original network, as we can see in the diagram. We have an original network that is on the left side, and it has been encoded into lower dimensional space with each node having a specific vector. And we use selective embedding techniques here for predicting missing links on gene regulatory networks using this representation learning or graph embeddings. Coming to the motivation, since we have the high throughput DNA microarray and most recent next generation sequencing technology for generating experimental gene expressions, but because of the lack of the true network, these links or networks, they're often guessed computationally. With a lot of progress in the field, we have a lot of unsupervised and supervised learning methods which have been developed so far, but they often suffer from a various type of limitations in capturing optimized network. So here is an alternative that we can predicting missing links for from our existing partial networks, which have been inferred through these techniques, in order to make these networks full-fledged, so that we can use in our real life world systems. And here are some of the contributions which have been outlaid here. We use some selective embedding methods for gene regulatory networks for predicting missing links. The first one is random walk-based method. And in random walk-based method, we use two methods. The first one is deep walk, and the second one is node to vec. These are two very popular methods, which also follow the network propagation rule in order to embed various for the various nodes of a graph. And the second one is a neural network based method, which we also called here as graph convolutional network, and it also has the name synonymous with graph neural networks. And we propose here an integrating framework for that can incorporate both gene expression values and gene network all together in order to do the missing link prediction. And we also evaluate here the performance of various embedding methods of various sized synthetic GRNs. And coming to the experimental setup of our paper, here on the extreme left side, we have the gene regulatory network, and it has a corresponding gene expression values to it, which are continuous in nature. And we do pre-process it, and this method we also say it has a discretization, and these values are actually converted into discrete values. And we also and when we bring these values together, we can also say these values as a feature matrix for all our gene nodes. And coming further, we have this main area of encoding, in which our adjacency matrix is given to the random walk based method. But our graph convolutional network, it takes both adjacency matrix of a network and feature matrix. So coming further, when we perform our technique and we get a lower dimensional space for each node, we get a lower dimensional space vector, which we also call latent space. And when we convert this into lower dimensional space, we also reconstruct our network with the sigmoid of the lower dimensional space with its transpose. And the output in the predicted network, we have these red links, which gives us the intuition of the new generated links in our gene regulatory network. And here are some of the results we have found, that the neural network based method, which is actually GCN, it has performed quite well for our various sized network types of ranging from 100 nodes to 4,000 nodes, our random walk based methods. And here are some of the trends that we could generate for various for various parameters of our GAE graph autoencoder or variational graph autoencoders, where we have got the good results. And here is a screenshot of our training and accuracy values for various data sets of our GAE and VGAE. And coming to the conclusion, we can see that the neural network based encoder, that is GCN, both for GAE and VGAE, it is superior in nature compared to the propagation based method for inferring missing links. And neural network based methods can capture the complex topological structure of the biological networks for various downstream tasks. And further, this work can be extended to various multi-layered heterogeneous biological networks. And the acknowledgement we give here is that this research is funded by DST, ICPS, which has been carried out at Natural Labs, Sikkim University. And here are some of the references. Thank you.